right, so we'll be moving on to the next lecture. That's lecture 2 and practical 1, which is Overview of Cryo EM Technique Workflow and Introduction to Instrumentation. This will be taken by Dr. Thomas Klose, still from Structural Biology, Purdue University. So I will quickly read his profile while he prepares to take the stage. So Dr. Thomas Klose has a diploma in biochemistry, as well as a PhD in biochemistry, which he had at the University of Halle-Wittenberg. He did a postdoc in genetics at the University of Halle-Wittenberg as well. He also did a postdoc and then was a research scientist in structural virology at Purdue University with Michael Roseman. He is the technical director of Purdue Cryo EM facility. He has more than 10 years of experience in cryo EM related techniques, and he has more than 70 publications, many of them related to cryo EM collaborations. Please, can we give a round of applause as we welcome Dr. Thomas.
Okay, thanks. I was trying to unmute myself, but that did not work. Thanks for the introduction. So as Wayne mentioned, and as was mentioned in the introduction, I am the technical director of the EM facility here since a few years now, and I'm going to try to give a more practical overview of what is done in cryo EM. There will be some overlap with what Wen just presented because theory and practice cannot be completely separated, but it will focus more on the practical workflow. So in very simple terms, what one does for a cryo EM workflow for single particle analysis, and that's what I'm going to focus on for now, is initially a sample preparation. Then once the samples are prepared, they need to be screened. Once screening is done, data will need to be collected, and then that data will need to be processed. So sample preparation is critical. However, if you look at the literature, you will find that some people say that it's less critical than for other structural techniques, because cryo EM is uniquely qualified to obtain structures of multiple specimens or conformationally flexible specimens or assembly states. However, even though we can computationally sort into different structures or different specimens, it is still advantageous to have a sample that is as pure, as homogeneous as possible to avoid downstream problems in processing and data collection. So our overall goal for sample purification would be to have a sample that only contains molecules that can be averaged together, so they're the same specimen, and or can be well separated from other specimens and contaminants. So while there have been some research projects that just take a cell extract and then try to get a structure from this that works kind of okay with ribosomes because they're very abundant in cells, but probably not with other molecules at this point. So once you have a purified sample, as Wen just briefly mentioned, the sample is getting applied to a grid that is exactly 3 millimeters in diameter and has a metal bar, which is what is the surrounding here, and then there are metal bars in the middle that go like this, and they form squares. And then on top of this, there is a support film. Depending on what you want to do and where you get your grids from, the metal bars are usually copper or gold. There are nickel grids available, but that's not advisable because nickel can be magnetic, and that can lead to interesting phenomena in the microscope because we're working with electromagnetism. The reason that it's either copper or gold is that both of them conduct heat very well, so we want to keep everything cold in cryo EM. So we want to transfer heat away and keep everything at liquid nitrogen temperature. And it also helps to distribute charge that eventually builds up during imaging. So we are going to bombard our sample with electrons, these electrons can create charge, and if there's charge in the sample that cannot go anywhere, then the image gets distorted because, again, that creates magnetic fields. And then the support film generally is either carbon or gold, and I'll show you in a bit more detail in a second, but the support film these days has usually defined holes with a defined spacing, and again, depending on the use case, this can be adjusted and selected. So most commonly what is done is holes that are 1.2 microns in size with a 1.3 micron spacing. If one has larger particles, one can go for a 2 micron hole with a 2 micron spacing. Or for really small particles, it's also not unusual to have a 0.6 micron hole with a 1.2 micron spacing or a 0.6 micron spacing. So again, which hole is selected depends a little bit on the specimen. Once you have your grid ready, the first thing that is usually done is what is called a Klose discharge. So these grids get manufactured and then packaged in a factory and then shipped to you. And over time, as they get exposed to the air, you get molecules like water or just impurities that are floating around in the air, and they like to stick to the carbon or gold layer of the grid, and they accumulate on this over time. So even though these are sealed and usually in a confined, like in a space that is humidity controlled and doesn't allow much airflow, there's still going to be stuff on there that you don't want. And there are also going to be slightly larger impurities, like there are going to be bacteria or viruses floating around in the air that you don't want. So the way to get rid of this is to apply what is called a Klose discharge. So essentially one takes the grid, puts it into an instrument that looks like this, establishes a pretty decent vacuum, and then there's a strong field that gets applied between two electrodes that creates plasma, and this plasma ionizes the molecules, they detach from the surface, and as a almost side effect, it also helps to make this carbon layer or the gold layer hydrophilic, which then helps, once we apply the sample to this layer, to actually spread it out, versus if it would be hydrophobic as it is out of the package, you would have problems getting the sample to spread out. So the droplet that would be placed on it would try to get to a minimal surface and not spread out evenly over the grid. And that is not really what we want later on. So once the grid is Klose discharged, we prepare the grid, and there are some annotations on my screen that are not from me. Not sure how to get rid of them. There we go. Okay. So what we typically do is that we take a few microliters of sample, depending on what your sample is, how much you have of it, how precious it is. The volume is typically around 2 to 4 microliters. Most commonly, it's right in the middle, like 3.5 microliters. And that gets applied to this grid that I showed you. So this is a side view of it, and it's held by tweezers here. And so what you end up with is a fairly large amount of liquid on this grid that is covering the whole grid surface. Then one uses a filter paper and moves this towards the grid surface. This takes away more than 99% of the sample, and as Sven also just briefly mentioned, we usually end up with a nanoliter or maybe a little bit more than a nanoliter left on the grid. And what we are aiming for is that we create a very thin water film, like kind of what is shown here, that is enough to support all molecules, but not so much that the ice becomes later on so thick that we cannot actually see anything. So it's typically in the nanometer to maybe half a micron at most range, but that is borderline for most microscopes. So we typically are below 500 nanometer or even below 100 nanometer for most samples. And then once we have achieved this very thin water layer, the filter paper is removed, and we are plunging into liquid ethane, and this allows us to cool the sample with a cooling rate of more than 100,000 kelvins per second. So it's really fast. And what we're aiming for there is to form vitreous or amorphous ice and preserving the sample in its native state. If we would cool much slower, we would get crystalline ice, and we do not want to have crystals in our samples. In this case, we don't want the ice to form an ordered state that would form any kind of diffraction pattern. And so that is the reason that liquid ethane is used. And then once this is done, the crits are transferred and stored in liquid nitrogen. So the reason that ethane is used is because it has properties that allow this really fast cooling rate, but it is actually dangerous. It's a flammable gas, and for safety reasons, you do not want to have a lot of it around. So once it is cool or has cooled rapidly, it is perfectly acceptable to have liquid nitrogen, which is roughly the same temperature, about minus 200 degrees centigrade. And that is a much safer storage place, because liquid nitrogen, if it evaporates, becomes nitrogen gas, which, unless it's really large quantities, is not really dangerous. So practically, how this is done these days is that you place your crit into a robot that looks like this. And this is what those of you that are on site will practice with in the cryo VR session. So this robot does exactly the same thing that I just explained to you. The robot has two arms here. There's a control panel here where you can change your settings, and then your crit gets transferred in here. These two arms come together, blot away the liquid, and then the sample plunges down into the liquid ethane that is going to be sitting here, and then gets rapidly cooled. So I do have maybe a video here. There we go. So I apologize if this is not playing correctly for you, but I will show links at the end where you can watch these videos on maybe a faster internet connection, or where you have more buffering time. So I'm going to mute this because I already told you this. So this is a crit that I showed you. And so we are slowly zooming in onto this crit. And so you can see as we get closer and closer, you can see the metal bars that I talked about. And there is this support film that has the defined holes in it. So as you get really close to these holes, you can see that in the holes there's ice, and in this ice there's your sample. In this case, I think they used a ribosome to demonstrate this. And this is how it is floating around in there. And so this is now a side view, and you see how your molecules are spread out in the ice. And then they're going to quickly show something that I will talk about later, which is the air-water interface. So this is how this looks in theory. And then I have a short video here that is going to show you how this actually works in practice. So again, those of you that will do the cryo VR part will be able to practice that in virtual reality tomorrow, I think. All right, so this is the cup that will hold liquid nitrogen on the outside here, and you will see that in a second. And then in the center of it is going to be the very small amount of liquid ethane that is actually going to act for freezing. So here they are adding liquid nitrogen to the cup. Now they're putting, with a very fine needle, the ethane in the center. So all of this is at about minus 200 degrees centigrade. Then this is the plotting robot that I talked about. So there are two filter papers that got mounted here. Now they are mounting the tweezers, which have the crit attached down here. And it gets pulled up into the robot and is sitting here. Then they will place the nitrogen freezing container down here. It gets lifted up really close to the chamber. The tweezers lowered a little bit. You open a port on the side, you go in with a pipette and you pipette your 3 microliters into the center of the grid. Then the grid will be pulled up. The plotting paper comes together like this for a defined amount of time that you can specify. And then they come apart and it plunges down into the ethane, and then the whole thing comes down. And then the most challenging part is to get the tweezers out from the robot, transfer it into the liquid nitrogen, and then the sample gets moved into a grid storage container that can then be stored in liquid nitrogen for a long time if nothing goes wrong. Okay, so this is how this works in practice. Once you have your grids frozen, what you need to do is screening. So screening is usually done on what some people like to call lower end microscopes. I don't really have a better term for it, but the idea is that it is rather challenging to find ideal conditions. So again, we want to have very thin ice, not too thick ice, but if we overdo our plotting, then we take too much liquid away and we have no ice. And while that sounds trivial, it can actually take months sometimes to establish the correct conditions. And so the reason that we use these lower end microscopes is that we can then use our higher end microscopes for data collection. And it is usually easier to get samples in and out of these less busy instruments. So what I mean by lower end microscopes is that they're usually at an acceleration voltage of 200 keV, or these days there are actually instruments on the market that are at 120 keV. And these acceleration voltages are sufficient for screening most protein samples. And they also are usually less expensive than the higher end microscopes. And one of the reasons is that they are a little bit less stable and less reproducible than the data collection microscopes. I mean, not by any large order of magnitude, but for quickly changing samples and just taking a few snapshots and trying to figure out if the ice is good and how the ice looks like on the grid, that is less of an issue. That being said, even these lower end microscopes are getting better and better, and they're actually very high resolution structures from what would have been considered a screening microscope a couple of years ago. So as I also just mentioned, our goals for these screening sessions are that we're getting an even and good ice distribution. We want to have as many squares with good ice as possible, because that will help us with data collection. We want ice that is as thin as possible. So there should be no excess water, but it should be thick enough to not force our particles to be at the air-water interface. And this is what was quickly shown in the first video that I showed you. When we plot the water away and it gets too thin, molecules like to go to the air-water interface. And if they do that, they will denature on the side where they're exposed to the air. And if you then take images of these molecules, they will be partially denatured on one side, and that is obviously not what you want. So let's say you have a molecule that is 10 nanometers in thickness. It is not a good idea to try to get an ice layer that is only 10 nanometers. You want to have a little bit of buffer at the top and bottom to prevent that your molecules will stick at the air-water interface. The other thing we want to achieve is that we have an even and as dense as possible distribution of particles in our ice layer. If they are not evenly distributed, it is very difficult to target good areas for data collection. And if they are less dense than would be ideal, then we're obviously going to have a hard time, or we have to take more images to achieve our target at the end. And then last but not least, we also would like to have random orientation of particles. And that is in theory easy, but some molecules like to orient themselves in the ice layer. And then one has to play tricks to get them to a more random distribution. So the way we do this practically is that the grids get either loaded into what is called a site entry holder, and that is typically used on thermal fishers like Techni or Telos systems. So the way this looks like is that this is your cryo holder. At the very tip of it, there is a shutter, and there's a space for your specimen, and then there's a very long barrel that is connected to a liquid nitrogen duo. So this gets filled with liquid nitrogen, and then it cools the whole thing down all the way to the tip. So it's kept at liquid nitrogen temperature. We take our crit and we put it right here. Then we put what is called a clip ring on top with this tool. This will hold the crit in place and also keep it cool because it is connected to the rest of the holder assembly. And then it can go into a microscope and can be imaged. On newer systems, and that is, again, from Thermo Fisher, it's usually a Titan Krios or a Glacios system. There's also a Joel system that uses a very similar approach. Things get loaded into what is called an autocrit, and that is shown here. So here you see this crit that I showed you before, over 3 millimeter crit that we have our sample on that we froze. And then this gets handled by a robot in the microscope. There is no robot that would be fine-tuned enough to really gently handle these very thin, fragile crits. So what we're doing is that we put them into a larger copper ring. Then the crit gets placed in here, and then we put what is called a clip ring on top. This clip ring sits in a little crevice around here and then really clamps down on this crit. So it is sitting in place tightly. Again, there's very good heat transfer. And then this assembly is much sturdier and can get loaded into what is called an autoloader, and then can be handled by a robotic arm. So again, I have a short video here. Sorry. So yeah, so this is the holder that I just showed you, and this was just set the what they call the workstation. So liquid nitrogen gets added on both ends here and here. This is now the workstation. This is the tip that I was talking about. This is where the sample will go. Here is your crit box that we use to put our sample in when we froze it. And you transfer it with tweezers into this opening. Again, this looks fairly simple, but it is actually quite challenging because your liquid nitrogen in here is heavily bubbling, as you can see. And so it's very easy to lose this crit when it floats away. So to prevent that, we put this clip ring on top, and this is what is shown here. And then this is what is called a TechNine. So this is the entry point for the microscope. It gets rotated, and then you will see how the sample holder comes in a second. There we go. So it gets inserted into the microscope. Wait a little bit, and then there's a second insertion step where, in this case, they're going really slow. And then the sample will be sitting in the center of the microscope column and can be imaged. So there we go. And yeah, we add more liquid nitrogen to keep it stable. And that's how the sample goes into a microscope in a side entry holder. For this autocrit, so this is what I just showed you for the automated systems, there is a dedicated workstation for that. So you put the bottom of the autocrit that I just showed you into a slot here. And for historical reasons, these Cryo EM crit boxes always have 4 spots. So whenever you freeze samples or prepare samples, you usually can do 4 at a time or multiples of 4. And so this workstation also has 4 slots to prepare the crits. Then this is how the clip ring gets inserted into the clip ring tool. So you take tweezers, you squeeze it into this little barrel, you orient it so that the clip ring sits at the very edge of this barrel. So you turn it upside down and push on it gently. And then you're good to go. So you cool all of this down. You put your crit box into one position. That is the crit box that has your frozen crits in it that you prepared. You open it up. Pick whichever grid you want. I guess they're going to go for this one. And then the grid gets transferred into the bottom part of the auto grid. Like this. So again, one has to be very gentle to not damage these grids, and the challenge is here that you're dealing with liquid nitrogen, which is minus 200 degrees centigrade. So you can't really get too close to it, otherwise it gets really cold and unpleasant. So you carefully position the grid in there. Then the top of this workstation gets rotated like this. And so now it is in a position where this clip ring tool can be positioned right on top of the auto grid. So remember, our little clip ring is at the bottom of this barrel. And so now put this on top, you push, and the ring will come out and expand and then hold the grid in place. So now they are going to rotate back and use special tweezers that are slightly differently designed to now take this grid assembly out of this position and put it into this second grid box that is over here. And then the sample is ready to be transferred to the loading station for the robotic system. And the robotic system can handle up to 12 grids at a time. So in theory, one could clip and prepare up to 3 of these boxes and 12 samples. So this is what is shown here now. This is the loading station. This is the cassette that will go into the microscope, and it has 12 positions here. So one takes the pre-assembled grid, puts it into this slot. They are checking that they are sitting at the right position there by gently pushing down on it, so it's all the way in and seated. And then the cassette, together with those auto grids, gets transferred into a shuttle that is called a nano cap. See that in a second here. Yeah, this one. So that obviously also needs to be cooled and has liquid nitrogen in it, because we want to keep our samples cold all the time. So it gets docked onto this loading station. The cassette gets lifted up and transferred quickly into the nano cap. And then that gets moved into the microscope, and this is what is called an autoloader. So it just push the nano cap in there, you push a button, it lifts up, and then the cassette gets transferred into the microscope. It will sit in here and is ready for you to load your samples. All right. Okay, so once you have it in the microscope, this is typically what you get. So we take low magnification images that will show us these squares that you've seen in the grid. And you can see that in this particular one, there's, for example, a big chunk of contamination here. So this is very dark and is not going to be able to image it. And then in this square, there is very thin ice on the edge here. So it's a very light gray. And then it's darker in the center. So that is thicker ice. And so it's a darker gray. So you would go probably to an area like this. You zoom in more, then you see your individual holes. So in this case, these are 1.2 micron holes with a 1.3 micron spacing between them. You find the ones that have thin enough ice, and you zoom in again, and you take another image at the sample level. And then you see here, in this case, these are ribosomes that are evenly distributed in the ice. And then if you have a grid like this would probably be ready for data collection. But as I said, usually takes a few attempts to get there. So what you're looking for is good ice. So as I said, we want to see our particles. They should be fairly evenly distributed, and we should see them. So our ice is thin enough to actually visualize them. And there should be no overlap between the particles. So like in this area here, for example, they are slightly aggregated, which is not perfect, but this is what I would call a good example. This is a different protein sample. And then in this third one, the sample concentration might not look ideal. There are quite a few empty spaces here, but for this particular case, sample prep is not easy, and one cannot get it to a high concentration because the protein would aggregate. So this is as good as it gets, and I would still say that this is a good distribution of sample in good ice. So what you don't want to see is something like this. So this, for example, is some kind of contaminant that was probably in the sample buffer, and then it froze, and then it looks like this. This cannot be imaged. Like that's not going to be good quality. This one here is an example of an uneven distribution of particles. So you see there's a large empty area here with no sample whatsoever. And then on the edge of it, the sample is actually slightly aggregated. So this would not be ideal. And then another thing that you don't want to see is something like this. So that is actually crystalline ice, which means that your sample either warmed up after you froze it. So your ice can actually transition from the amorphous state that we want to a crystalline state if it gets above about minus 150 degrees centigrade. And then this is not going to be good for imaging either. This will not allow us to see our particles, and it produces these like weird artifacts. So once you have established a good ice, you want to go ahead and collect data. What we do for this is that we prepare the samples and load them in the same way as I just discussed. So there's no difference between a data collection session and a screening session in terms of how the samples get into the microscope. What we usually want to do is that we have multiple crits with same or the similar condition in the microscope at the same time. The reason for that is that it is not a very precise science to freeze these samples. So even if I give you the same sample and you freeze the crit, like you freeze 4 crits with the exact same conditions, they are not going to look exactly identical. And so because of these small variations in crit quality, it is good to not just have one crit in your microscope, but to have something that you can choose from and then pick the best crit. And that's what we then usually do. We do very minimal screening on the day of data collection. So we go through those crits, we look at them at a very rough overview, we find which ones have good ice, maybe take 1 or 2 snapshots at high magnification to make sure that the protein is still there. And then when we have identified the best candidate, then we set up data collection. And at this point, these typically run overnight. So we start like sometime early in the morning, we do our minimal screening that takes about 2, 3, 4 hours. Once we have our crit, we set everything up, and then it runs for another 20 hours. And that is enough data to get a structure. So for data collection, we do have imaging states. So as I said, we do take, again, an overview of the crit at a very low magnification where we can identify good-looking squares. Then we zoom into these squares, and it will look like this. In this case, again, we can identify areas that we like. Then we zoom in even more and identify holes that look good. And then at the end of it, one takes a snapshot in this hole and sees the sample, which again, like this is a very good distribution of sample in the ice. And it's like probably hard to see, but there are lots of tiny protein molecules there. And then you repeat that many, many times over until you have thousands of images taken. From a user point of view, what you want to know, or what you will probably be asked when you set up the microscope, is what magnification you want to use. And that translates to what pixel size you will have. As Sven said in his talk, the maximum achievable resolution is limited to twice the pixel size. So let's say you are being ambitious and you want to have a 2 angstrom structure, your pixel size needs to be 1 angstrom, or actually smaller than that, because we can, like, almost nobody has ever achieved the maximum possible resolution, like theoretically possible resolution, because the signal to noise ratio is just not very good when you get very close to the maximum. Obviously, larger pixel sizes, so zooming out more, give you a bigger field of view, and that will give you more particles per time unit. But the more you zoom out, the more you're going to be limited in your resolution. So one has to have a realistic idea and goal for what resolution one wants to achieve, or if it is better to zoom out a bit more and get more particles because they are a mix of states or something like this. The next thing one needs to think about is the defocus range. If you're close to focus, images have very little contrast, and it's very difficult to see the particles, but the signal in the actual micrograph is preserved at the best way possible. And if you have large defocus images, they have very high contrast, makes it very easy to see your particles. But there is what is called signal delocalization, and I think Wen mentioned that as well. And that actually will not help you get the highest resolution structure possible. So one has to strike the balance between actually being able to see your particles and preserving as much signal as possible. And like, yeah, not getting so close to focus that the particles disappear. And practically speaking, this range is usually between 0.5 micron to about 2 microns defocus. And then last but not least, one has to know what dose you want to apply. All biological specimens are very beam sensitive. So when we are bombarding them with electrons, they get destroyed. So you can actually see them blow up over time. And so any kind of beam exposure will actually lead to damage, but we can compensate for some of this damage during data processing later. And so most biological specimens can tolerate up to 120 to 200 electrons per angstrom square. And so practically speaking, what most people collect data with is about 40 to 60 electrons per angstrom square. And then the other consideration there is that to get more dose, one needs to expose the image for a longer time. So if you want to have as many images as possible per time unit, it is actually better to be on the shorter dose range, but you have to make sure that it's enough dose to see your particles. So these are things that one should discuss with a facility staff member or so before data collection starts. And these are the questions that typically come up. So this is just to give you a quick example of magnification. So this is the very same sample at a fairly lowish magnification. You can see many particles, but you don't see many features in there. Then this is like a medium high magnification. There are obviously much fewer particles, but you can actually see many more details. And then this is a really high magnification, but you're only ending up with like 7, 8 particles in the field of view. And this might be now overkill to actually get what you want. So for high-ish resolution structure, one would probably go with this magnification. And if you want to go really high resolution, maybe with this magnification, but this is probably not practical because it would be too few particles and it would take forever to collect the data. And this is what I was talking about in terms of defocus. So up here, you have a high defocus. So this is a 3 micron defocus image. You can hopefully see the particles fairly clearly. But you can see that if you look at a Fourier transform of this image, where the lowest resolution is in the center and then high resolution information spreads out to the edge, you only see information to, let's say, roughly here. So it is very limited in terms of what information you can just recover. Whereas if you go to a low defocus image, it is much more difficult to see the individual particles, but you can actually see that the information is there to a very high resolution. So this would be borderline actually seeing particles. And so what one ends up with is usually a happy medium. So this is at 1.5 microns defocus. One can still see the particles fairly well, and information spreads out almost as far as it does for the very low defocus image. So again, these things need to be balanced. And what we typically do is that we vary the defocus during the data collection between roughly the 0.75 micron and the 1.5 micron, but we would not go to a 3 micron defocus because there's just not much information left when we process the data. Then you also have to think about how you target your image. So we have all these holes and we take a snapshot in each hole. The traditional way of doing this is to move the microscope stage to the center of each hole, then take an image, and then move to the center of the next hole, take an image. That is very slow because you have to move every single time, but it can be very precise, or that's probably the most precise way of targeting. It has the benefit that the microscope is perfectly aligned because the beam comes straight down and is not getting interrupted. And if you use this targeting technique, which was state of the art until a few years ago, you can expect less than 2,000 micrographs / movies in a 24-hour session. That is not really sufficient for today's standards. So what is more typical these days is what is called an image beam shift after stage move. So what you do is to move your stage to the center of a bunch of holes, and then you can shift your beam to target individual holes. So in this case, 4 of them. The drawback of that is that you're moving the beam off the central axis of the microscope, and that induces what is called coma. However, these days we are able to predict how much of this coma gets induced and actually correct in the microscope for this coma effect. And that is so good that it is essentially non-existent for practical purposes. So if we do this, we can increase our throughput to 3,000, 4,000, 5,000 movies in a 24-hour session. So that is a factor of like 100% more than you would get with the very slow targeting mechanism. And then if you push this to an extreme, and this is actually what is done most of the times these days, is that you're not only taking 1 image per hole, but you, in this case, take 4 images per hole. So you still move your stage to the central position, and then you can take, instead of 4 images, you can take 16 images. The drawback, again, the drawback would be that there is this off-axis coma, but again, we can compensate for that very easily. The only thing one really has to pay attention to is that if you take an image here, this image cannot overlap with the neighboring image, because otherwise this area would see twice the electron dose, and that would lead to damage in the molecule. So one has to be very careful to lay that out so there's no overlap between the exposure areas. But if you do that, we can actually, under ideal circumstances, achieve about 12,000 movies these days in a 24-hour session. And that is obviously very good if you want to have as many molecules as possible. Okay, so once you have all your data, you need to do data processing. And the rough steps for that is that you import your data, then you do what is called motion correction, then CTF estimation, then you pick your particles and extract them from your micrographs. Then you do what is called a 2D classification, an initial model, you refine, and then you need to validate and build your model in an iterative procedure. And Daisuke's group will talk about more of this tomorrow. So I'm just going to quickly go over these steps. And if you're interested in that, there are many tutorials in the web how to do this that are fairly accessible. So I don't want to repeat something that you can easily read up on. So the first step, as I said, is to import your data and do what is called motion correction. So what we do these days is that we we record our data as a movie. The reason for that is that when we put the beam on our sample, this leads to motion of the particles in the ice layer, and it actually also leads to deformation of the ice layer itself. So you get what is called a doming effect. So your ice would be fairly flat in the beginning, and then it would like kind of start to raise up slightly. On top of that, we are dealing with mechanical movement. So when we move the stage where the grid is around and we stop, there is going to be a little bit of drift. And so if you ever have tried to take a picture of a cat or a dog that is running really fast, you will know that it's almost impossible to get a good picture. It's going to be blurry because your object moves as you take the picture. And so to compensate for that, we instead take many, many short duration pictures and combine them in a movie. And then we can use an algorithm that compares each frame of this movie with the previous and the following one. And then it can compute a trajectory to bring them in alignment and essentially eliminate most of the motion that was experienced. So an example of that, and this is probably a little bit difficult to see on the screen, and the easiest is down here, but here's a unaligned movie stack just averaged together. And if you look very carefully at this, you can see that these particles are ever so slightly blurry. And if we do, again, a Fourier transform of this, which shows us the resolution of the image, so again, this is the very low resolution, and then we go to higher resolution, You see there's some small ripples in this direction, but all the high resolution information in this direction is lost because of the image was drifting downwards as it was taken. So after we do this motion correction, we can actually see that the particles are much more visible, very well defined, and resolution information has been restored in all directions. So this is what we want. And then in addition to all of these things, as I said, any beam exposure will cause damage to the particles, and that will lead to loss of high resolution information. But over the years, we have learned when particles take damage. And so we can actually apply what is called dose weighting to compensate for this beam damage. So essentially, in the later part of the movie, we are not considering high resolution information in the average anymore, because that high resolution information was already lost. And we only retain the high resolution information from the early parts of the movie where it was still present. So once you've done this, you have to do what is called CTF estimation. So essentially all that this does is to accurately determine the defocus that was applied during the exposure. So I told you that you have to decide what your defocus range should be, and your microscope will set a certain defocus, but that is never 100% reliable. So our nominal defocus cannot just be used to be applied to the image, and we need to correct for the effects of this defocus. So we need a very precise estimation of the defocus value. And so what the program essentially does is to, again, do this Fourier transform, and then it can, based on how these ripples are spaced apart, it can determine what the defocus was. And then this information gets saved and then gets corrected for during downstream processing. All of that is done automatically. You usually don't have to think about it much. Then you go and do particle picking. So particles need to be identified in each micrograph, and then you need to extract them from the micrograph. The typical way you do this is these days you can do it based on blobs. So if you look at a particle at very low resolution, they resemble essentially blobs. And so if you know the rough dimensions of your particle, you can just generate a blob that is the right size, and then you can use an algorithm to find correlation between that blob and your micrograph, and that will fairly accurately pick your particles. The slightly more advanced method of this is to use a template. So that would be an actual image of your particle that you already have, and then you can use that and search your micrographs for additional copies of that particle. Or the most modern approach is that you have artificial intelligence models that are trained on either your data set, so you train it yourself, or you can sometimes get away with like networks that are trained on similar protein structures. And what I'm trying to show here is that this is your raw micrograph, and this is the micrograph where all the high resolution information is removed. And so this is what is called low pass filter to 20 angstroms. So everything higher than 20 angstroms is not present in this image anymore. And so you see, if you do that, the shape of the molecule is preserved, but the high resolution features are gone. And that eliminates a lot of noise between the particles and makes them easier to pick for these algorithms. So either if you do it manually or if you use any of these methods to pick your particles, it's what most of them do is to remove all the high resolution information just for picking and finding the particles. And then once you have identified all your particle locations, you need to extract them with a box around them. And in very simple terms, this box obviously needs to cover your particle. So it needs to be as big as your particle. But we also need some buffer around it because of this defocus that I was describing earlier. There's some of the high resolution signal gets delocalized from the center of the particle. And then if you or the algorithm is not 100% perfect in identifying the center of the particle, you also want to have some buffer so that your particle can move around and there are imperfections in determining the center of the particles. So practically speaking, what you usually do is to take a box that is about 1.5 times as big as your particle. Then when you've done this, you end up with boxes that look like this. So you see it as the picking obviously picks particles, as it should, but the picking will also pick contaminants that look roughly like your particle, or weird little blobs on edges of contaminants. Or in this case, this protein was probably denatured, so it looks actually very bad. And so what you want to do is to do a 2D classification. So the program will look at all these individual particles and combine those that are similar and then give you an average. So in this case, these are all 2D views from your micrograph of your protein, and it would put these contaminants into a separate class and you can easily sort them out. Once you have all these 2D views, you can then combine them into a 3D model, and Wen talked about this. So you essentially combine them together, and if you do that in the right way, then you can get a very low resolution model. So it will look like something like this at 40 angstroms. Then you can take this model and project it into a 2D view again, and then you compare your actual particles against this 2D view, and then this way you can refine their position and orientation in 3D space. And you do that, and then you will get to a slightly higher resolution. So this is the same molecule at 10 angstrom resolution. So you can start to see pretty much the detailed structure, like where individual domains are and even how the molecule is shaped. And then if you keep doing this and your data quality is good and your protein quality is good, you can get to like a 3 angstrom or even better structure. And again, this is highly automated these days. So you usually push a button and you can follow it along and it will give you a nice structure at the end. So with that, I'm at the end of what I wanted to talk about. So as I said, there will be another part of this workshop which will talk about validation and modeling. So I'm not going to go over that, and I'm happy to take any questions.
All right, the online participants, I hope you can hear me now. I thought that was an awesome lecture by Dr. Thomas Klose. We were not connected, so can we clap again so that they can hear? All right, thank you very much. That was comprehensive and explanatory, and it really builds on the background foundation that Prof. Weng Jian had already established in the first lecture. And some of us had the privilege of doing this virtually yesterday. We saw a little bit, we saw a glimpse of it, entering into the virtual environment to do this yesterday. And I believe we could relate. So we are still going to have more of that during the break and tomorrow as well. So we're going to be performing the experiments in the virtual environment. So I would encourage those who are online and you're in the city of Jos or you're close by, please do come so that you can have hands-on experience with this experiment. And then for those who are online, everybody's going to have the opportunity to enter into the virtual environment to do this. So right now we'll be taking questions. Please, do we have questions for Dr. Thomas so that we can, so for those on site, please, anybody with a question? All right, we have someone with a question, Dr. Simeon O'Malley. So you can ask a question.
Very much, Thomas, for the extensive lecture and
demonstration. I'm having a very hard time hearing the question. Like there's a lot of background.
Okay, let me put that to the mic. I was saying thank you very much for the extensive lecture. That's awesome. I must confess, it's a little bit advanced for us, but in one of the slides, I saw the pathologies when you were trying to view your results, and one of it was contamination, another one was uneven, and I think the third one was, is it crystallized or something? So I was wondering if issue of contamination is a concern then the process that we saw in the demonstration was done with bare hands and all that for me. Is it, at what point does this contamination take place? Because I saw the demonstration when the samples were loaded. I am not seeing too much of caution. So at what point should one be careful of contamination using this cryo EM?
So you should be careful at all points, right? So what they showed in the video, so there's a website that is supported and funded by the same NIH grant that is also enabling the cryo EM. So it provides the background and everything from that, and that's where I took this video from. And we typically use gloves to freeze and prepare liquids, but that being said, the kind of contamination that I showed you, that's usually in buffer or something like this. So I would say if you use good standard practice of like filtering your buffers and keeping your samples clean in general, then you should have very little problems with the first type of contamination that I showed. So that is, it's usually a sample impurity, let's put it this way. So like, and the other thing that one has to keep in mind is that we frequently get people that prepare their sample, they have a very nice peak on a size exclusion or something like this, and they say, my sample is 100% pure. But when we are freezing it, we see individual molecules, and you're obviously not going to pick that up in a bulk purification. So even if you did your very best job, you're going to see some contaminants that are not, that you wouldn't expect. So as long as there are only a few of them, you're fine. The second kind of pathology that I showed with the uneven distribution, that is a matter of optimizing. So as I said, the process of freezing liquids, you usually have to do multiple rounds of it. And so what I showed there, it's very likely that on what was on the left-hand side of the image, the ice was just too thin. And so the particles were not able to get into that space because there was just no space for them. So that could likely be corrected by changing the plotting time, so taking away a little bit less liquid, and so having slightly thicker ice that then would allow the particles to get in there. The third pathology that I showed, like the crystalline ice, that is usually a user error. So when you're freezing liquids, everything that the grid gets in contact with during freezing and after freezing has to be at least minus 170 degrees centigrade. So every tool that you're using, everything that you're doing with the grid needs to be cooled in liquid nitrogen, and it needs to be cooled until it's really cool. And so the crystalline ice we actually frequently see with beginners that are a bit nervous or not patient enough in like cooling their equipment down, and then they warm up their grid. And again, like I know it's for many people that's counterintuitive that minus 130 degrees centigrade is warming up, but it is enough to transition to crystalline ice, and then you get this formation. So the most common reason for seeing that is that the cryogen in which the grid was plunged was not cold enough, and that is fairly easily corrected. Like you can just wait a little bit longer and make sure that your cryogen is really cool enough. So yeah, like I know that all this information is overwhelming, and it's probably not so useful until you actually see it, but it is it's important to kind of keep that in mind, that it's not just freezing a grid and then everything is fine. There are things that can go wrong after that.
All right, thank you very much, Dr. Thomas, for that. We have another question. I'll be taking this from the online participants. How to know when to stop the refinement? Like, how do we know when to stop the refinement?
Most people never do. So it's very difficult to answer that question because technology is still advancing a lot. So practically speaking, what most people do is to put their data into one software package, and then the software package will actually say like, okay, I can't do any better with the data that you gave me. And then it will terminate the refinement. If you are satisfied with that result, then most people just let it be. But then if you're not, there are usually many settings in these programs. They're very advanced, and that would like just be too much for this workshop to go over them. But there are certain things that can be refined in addition to the usual typical refinements that sometimes helps. And that's actually where Ben's group has done a lot over the years to correct for aberrations or distortions that might be in the microscope. But if that all doesn't help and one is still not satisfied, then usually one has to go back to step one, get new sample and get better sample or more images. The practical consideration is really like, what do you want from your data? If the goal is that you want to identify a very small ligand in an enzyme, then you can probably, and you reach below 2 angstroms resolution, you can probably be happy and you will get your biological information that you need and be done with it. If you just want to see what kind of complex your protein forms, like even probably at 3.5 angstroms or even maybe 4 angstroms, one can get this information. And so this is what most of our users do. They kind of stop when they get the answers that they wanted, and the program tells them that they can't get any higher. But there's no surefire way of knowing like, this is really what my data was able to achieve and it doesn't go any better. It's a lot of try and error, and you can spend years refining it.
All right. Thank you. I believe that Chandan Vishwakarma was able to get that explanation. Another question, which is going to be the last before we take the break, is how to deal with aggregate formation in case of membrane proteins to be used for cryo EM. How do we deal with aggregate formation in case of membrane proteins to be used for cryo EM?
There are many different approaches, and again, that will very much depend on what it is. The most common ones are to use detergents, like mild detergents, because most membrane proteins aggregate with their transmembrane part that is hydrophobic, and that hydrophobic part likes to stick to another hydrophobic part. So there are actually detergent screens available nowadays for cryo EM that can be added to membrane protein preparations, and then one can see which one works best. There is, again, it's not that I can tell you have to use this one and it will work. You usually have to try a few and see what works best. Another very common approach is to put them into nanodiscs. So there you essentially have a protein scaffold that can accommodate lipids, and then your membrane protein gets embedded into its native lipid environment, and the scaffold around the nanodisc provides enough buffer so that the membrane protein cannot aggregate with the next membrane protein. So and then some people go to the most extreme thing, that they actually add liposomes and then embed their membrane protein into these liposomes and then collect images of the membrane protein in a liposome. So I would say these are the 3 most common ways of avoiding aggregation. But again, this needs to be tested. So, and practically speaking, membrane protein projects are the ones that spend the most time in the screening stage, because it is so difficult to get the protein to begin with, and then it is really difficult to get a fairly good distribution of the particles that are not aggregating and are monodisperse.
All right, thank you so much, Dr. Thomas Klose. We really appreciate your time and the knowledge that you have been able to pass on to us. We will be going on to the break right now. We've made a little bit of adjustments to the schedule. So our break is going to be lasting for about 50 minutes instead of 30, as it was meant to be. So for our online participants, you can take your break However, you can follow what's happening on site as well. And for the on-site participants, you can have those who want to go on break but we're going to be starting a practical hands-on experience on the virtual reality right now during the break. So thank you very much.
Before we go on break, can we please appreciate our facilitators once more, please? Those online, please, you can say some chat, you can send some chat to appreciate Dr. Thomas, and also to appreciate Professor Wenjiang, who just gave lecture a few minutes ago. So thank you very much. We will have a break and we'll be coming back by 4:50 Nigerian time. 4:50 Nigerian time. So it's not, that's not up to, how many minutes should that be now? 4:50. This is 4:17 right now. About 30-something minutes, right? 33 minutes, okay. So in 33 minutes, we should resume back the next session. The facilitator will be speaking also on Cryo VR, what we're already setting up over there. It will let us know what is used for the relevance as it relates to Cryo EM that has been introduced to us. And we also have, some of us, we have hands-on today on it. Then we have sessions tomorrow where the remaining of us will have further hands-on on it if all of us can't have it today. Again, we will, in the break, if you're on site and you're not going anywhere, we'll show you how to create accounts for this Cryo VR stuff. For those online also, we still have the session again, but we can start putting those on site through. This is how to create the account and also how to log into our website, the Cryo VR website, to download the modules that we have and also to install it on your on your Oculus headset if you have one. Jahwe also will be talking more about that in the next section for those following online. So you're not missing out anything if you're following online. You will also have everything set to you. Okay, so without further ado, we will take a break now. The Zoom link, the Zoom will be on. So if you want to stay on Zoom, you can stay. If you want to take a break, you can take a break. For those on site also, if you want to stretch your legs, take a break, you can do that. But we are still here to continue. Thank you once more for being here.